Every mitochondrion in your body is being measured right now. Cell is running a continuous voltage test across the inner membrane of every single one, checking whether each mitochondrion is maintaining its 180 millivolt charge. Ones that are failing are being flagged, engulfed, and dismantled. Their components are being recycled into new structures. This is happening in you at this moment as a background process that never stops. Fasting does not start this process, it accelerates it. Dramatically, measurably, at the molecular level, in a way that the constantly fed state suppresses. The Nobel Prize in 2016 was awarded for describing the mechanism. Almost nothing explained since has done it justice at the level of physics. There is a threshold, approximately 16 hours into a fast, where the cell undergoes a shift that is not gradual but discontinuous, a phase transition in the physics sense from growth mode to repair mode. There is a voltage-based quality control system that identifies failing mitochondria by measuring their electrical charge and dispatches recycling machinery to their precise location. And there is a molecule, beta-hydroxybutyrate, that fuels the brain during fasting while simultaneously reprogramming the brain's gene expression toward growth and plasticity. All of it comes later. Start with why the cell needs to eat itself at all. Because the answer is the second law of thermodynamics. A living cell is a thermodynamically improbable object. It maintains precise order, specific ion concentrations on specific sides of membranes, proteins folded into exact three-dimensional configurations, organelles positioned and functional against the continuous pressure of the second law, which drives every physical system toward maximum disorder. This maintenance is not passive, it requires continuous energy expenditure. Sodium-potassium pump alone consumes approximately 30% of a cell's resting ATP budget, simply to maintain the ion gradients that make neural signaling and cellular function possible. Proteins misfold continuously. Thermal noise alone causes conformational errors at a measurable rate and must be detected, refolded, or destroyed before the misfolded copies aggregate into structures the cell cannot process. At body temperature, a protein with 300 amino acids experiences sufficient thermal vibration to sample incorrect conformations thousands of times per second. Most of these are corrected instantly by the protein's own thermodynamic landscape. The energy minimum that defines the correct fold pulls it back. But a fraction escape into stable, misfolded states that the protein cannot correct on its own. These must be caught by chaperone proteins, refolded, or tagged for degradation. If they are not caught, they aggregate. Misfolded proteins bind to other misfolded proteins through exposed hydrophobic surfaces that should be buried in the interior of the correctly folded structure. The aggregates grow. They become resistant to degradation. They accumulate. Mitochondria accumulate oxidative damage from the reactive oxygen species. Their own electron transport chains produce as byproducts of normal operation. Superoxide, hydrogen peroxide, hydroxyl radicals that damage mitochondrial DNA, mitochondrial membranes, and the electron transport chain, proteins themselves. Membrane lipids oxidize. Nuclear DNA sustains lesions. Every ordered structure in the cell is being degraded by thermodynamic noise at every moment, and the cell must spend energy to repair or replace every one of them or lose function progressively. Abundance changes the balance. When food is plentiful and ATP is plentiful, the cell runs maintenance alongside growth and synthesis simultaneously, repairing damage while also building new proteins, new membranes, new organelles. The growth signal dominates. The maintenance runs in the background. The balance favors construction over repair. In scarcity, when food stops and ATP becomes constrained, the cell faces a resource allocation problem that evolution solved with remarkable precision. Instead of running at reduced capacity across all systems, it switches modes entirely. It begins generating energy by consuming its own damaged components, recycling the molecular debris that entropy has been accumulating, and using the recovered building blocks to maintain its highest priority functions. Growth stops. Repair accelerates. The balance inverts. Cell does not starve during a fast. It audits itself. Think about that. The second law of thermodynamics, the most fundamental law in all of physics, is the reason cells need autophagy. Entropy is not a background condition of the universe. It is an active force acting on every protein, every membrane, every organelle in every cell in your body, every second of your life. Fasting is the condition under which the cell mounts its most aggressive response to that force. Now, the switch. Because the shift from growth to repair is not a dial, it is a phase transition. Water does not gradually become ice. At a precise temperature, it undergoes a discontinuous change of state, liquid on one side of the threshold, solid on the other. The transition is not a slow stiffening, it is a boundary event. The mTOR system that governs autophagy operates the same way. mTOR, the mechanistic target of rapamycin, is a protein kinase that acts as the cell's primary nutrient sensor. When amino acids are abundant and growth signals are present, mTOR is active and suppresses autophagy completely. The cell is in growth mode, building, synthesizing, proliferating. Autophagy is held at baseline. When amino acid levels drop below a threshold, approximately 12 to 16 hours into a fast in humans, AMPK, the cellular energy sensor that monitors the ratio of AMP to ATP, crosses its own activation threshold. AMPK inhibits mTOR. Inhibition is not partial. Inhibition is not partial, it is a switch. Below the threshold, autophagy is suppressed. Above it, the brake is released and autophagy initiates across the cell. This is why the difference between 14 hours and 16 hours of fasting is not proportional. 14 hours may produce almost no autophagic induction. 16 hours may produce significant induction. The physics of threshold systems means the benefit does not accumulate linearly. It arrives at a boundary, and the boundary is the mTOR to AMPK switch, integrating signals from amino acid levels, growth factors, oxygen status, and energy state simultaneously. 1 protein kinase. 4 inputs. 1 binary decision about the cell's operating mode. The person who eats their last meal at 8 in the evening and their first meal at 10 the next morning has fasted for 14 hours and may have produced minimal autophagic activation. The person who waits until noon has fasted for 16 hours and may have crossed the threshold. The 2 additional hours are not 2 additional hours of gradual benefit. They are the difference between one side of a phase boundary and the other. The physics is discontinuous. The popular fasting content that treats hours of fasting as linearly beneficial, implying that 12 hours is good, 14 is better, 16 is best, in a smooth progression, is misrepresenting a threshold system as a linear one. The benefit does not scale smoothly. It switches on. Well, and the man who discovered the mechanism was not studying fasting at all. The mechanism was discovered not in human cells, but in yeast, single-celled organisms under nitrogen starvation. Yoshinori Osumi was watching something nobody had examined carefully before, the yeast vacuole, the organelle equivalent of the mammalian lysosome. Under nitrogen deprivation, the vacuoles filled with small membrane-bound bodies he could not identify. He engineered yeast strains that could not degrade the contents of their vacuoles, so the bodies accumulated visibly. He was looking at autophagosomes, the membrane structures that engulf and deliver cellular debris for degradation. He had found the threshold in yeast. Over the next decade, he identified the genes responsible, the ATG genes, autophagy-related genes, and mapped the molecular machinery that builds the autophagosome, targets its cargo, and delivers it for degradation. The work was painstaking, conducted in an organism most molecular biologists were not studying, on a process most cell biologists considered a minor housekeeping function. Threshold. He identified operates in your cells. The molecular switch is conserved across more than 1 billion years of evolution, from single-celled yeast to every cell in the human body. The ATG genes he identified in yeast have direct human homologs that perform identical functions. The machinery is the same. The trigger is the same. The physics is the same. The physics is the same. He received the Nobel Prize in Physiology or Medicine in 2016 for describing a process that had been running in every eukaryotic cell since before multicellular life existed, and that nobody had examined carefully enough to understand. Now, what happens after the switch flips? Because the structure the cell builds in response is a piece of engineering that is almost never explained at the level of physics. Autophagy begins with the formation of an autophagosome, a double membrane vesicle that engulfs damaged cellular material and delivers it to the lysosome for degradation. The physics of how this structure forms is extraordinary. A lipid bilayer membrane has a natural resting curvature of 0. It prefers to be flat. Curving a membrane away from flat requires energy, described by the Helfrich bending energy equation. The energy cost is proportional to the bending modulus of the membrane and the square of the curvature imposed. To form a closed spherical vesicle large enough to engulf a mitochondrion, which may be several micrometers in length, the membrane must be bent into a high curvature geometry against its energetic preference. Flat membrane does not want to become a sphere. The cell forces it. ATG proteins, Osumi identified, do this mechanical work. Specifically, the ATG12, ATG5, ATG16L1 complex, and the LC3 lipidation system work together to sense membrane curvature, recruit lipids to the growing phagophore, the expanding sheet of membrane that will become the autophagosome, and drive the bending and closure that produces a sealed double membrane sphere. The entire structure assembles in approximately 10 minutes, from flat lipid precursor to closed autophagosome, large enough to contain a damaged organelle. 10 minutes. Membrane doing mechanical work against its own physics, building a structure from scratch, guided by proteins expressed specifically in response to a nutrient signal. This is not a metaphor for cellular efficiency. It is Newtonian mechanics operating at the nanometer scale. You see, every 10 minutes that a fasted cell is running autophagy, it is completing a full construction and recycling cycle. Membrane recruited, curved against its energetic preference, sealed around a target, fused with a lysosome, contents hydrolysed by lysosomal enzymes, building blocks, amino acids, lipids, nucleotides, returned to the cytoplasm for reuse. The cell is running a manufacturing and recycling operation simultaneously, and the entire operation is powered by the physics of membrane bending and protein-lipid interaction triggered by the energy signal of a fast. Single cell may run dozens of these cycles simultaneously during active autophagy. Multiple autophagosomes forming, engulfing, fusing, and degrading at overlapping stages. The throughput is substantial. In a few hours of active autophagy, a cell can turn over a significant fraction of its damaged protein and organelle inventory, replacing dysfunction with recovered building blocks that are then reassembled into functional structures. The cell that emerges from a fast is not the cell that entered it. It has audited its inventory, recycled its damaged components, and rebuilt with the salvaged material. The cell is cleaner, younger in its functional capacity, and running on a mitochondrial population that has been pruned of its weakest members. And then, the part that changes what the process means. Because autophagy is not random, the cell does not eat itself indiscriminately. It targets the most damaged components first. Most important recent discovery in autophagy research is selective autophagy, the cell's ability to identify specific damaged structures and target them for degradation while leaving healthy structures intact. The clearest example is mitophagy, the selective autophagy of damaged mitochondria, governed by the pink-1-trockin system. Governed by the pink-1-trockin system, pink-1 is a kinase that is continuously imported into healthy mitochondria and degraded inside them, keeping its concentration on the outer mitochondrial surface near 0. This is the default state. pink-1 arrives, enters the mitochondrium, is destroyed. The healthy mitochondrium is invisible to the autophagy machinery because it has no pink-1 on its surface. When a mitochondrion's inner membrane potential drops below threshold, when the 180 millivolt charge that drives ATP synthesis weakens, indicating dysfunction in the electron transport chain, The import machinery that pulls PKK1 into the mitochondrion stops working. Import requires the membrane potential to drive the translocation. Without adequate voltage, PKK1 cannot enter. It accumulates on the outer mitochondrial membrane instead. Accumulated PKK1 phosphorylates ubiquitin molecules and recruits parkin, an E3 ubiquitin ligase, to the outer membrane. Parkin ubiquitinates multiple outer membrane proteins, creating a dense molecular flag visible to the autophagy machinery. Autophagy receptor proteins, P62, NDP52, OPTN, recognize the ubiquitin chains and recruit the growing phagophore directly to the flagged mitochondrion. The autophagosome forms around it specifically. The damaged mitochondrion is engulfed, delivered to a lysosome, and degraded. Its components are recycled. Cell identified a failing power plant by measuring its voltage. It flagged it through a molecular cascade triggered by the electrical failure. It dispatched recycling machinery to the precise location. Every mitochondrion in your body is under continuous voltage surveillance. The ones that drop below threshold get removed. Elegance of this system is in what it does not require. It does not require the cell to chemically analyze each mitochondrion's output. It does not require a census of mitochondrial DNA damage or a protein-by-protein audit of the electron transport chain. It uses one measurement, voltage, as a proxy for overall mitochondrial health, because a mitochondrion that cannot maintain its membrane potential cannot be performing its primary function. The measurement is fast, continuous, and binary. Above threshold, the mitochondrion is invisible to the recycling machinery. Below threshold, PKK1 accumulates and the mitochondrion is marked for destruction. The entire quality control decision, keep or recycle, runs on a single electrical parameter. In a continuously fed state, with mTOR active and autophagy suppressed, flagged mitochondria accumulate. The voltage surveillance identifies them. The PKK1 marks them. But the autophagosome machinery that would engulf and recycle them is running at reduced capacity because mTOR is holding the brake. The damaged mitochondria persist, producing less ATP, generating more reactive oxygen species, accumulating further damage. Fasting releases the brake. The autophagosome machinery activates. The backlog of flagged mitochondria is cleared. New mitochondria, generated through mitochondrial biogenesis, driven partly by PGC1 alpha, a transcriptional coactivator activated by AMPK during fasting, replace the ones that were removed. The mitochondrial population after a fast is younger, less damaged, and more efficient than the population before it. That finding is what made the fasting research land differently for me, because it means the cell is not making a chemical judgment about mitochondrial health. It is making an electrical one. The physics of membrane potential, electrochemistry, identical to what drives nerve conduction and muscle contraction, is the instrument the cell uses to identify which of its power plants need to be replaced. The quality control is electrical. The measurement is voltage. The consequence is recycling. And here is the layer that most fasting content never reaches. Because the identity of mitochondria adds something to this that changes what the quality control means. Mitochondria are not native cellular structures. They are endosymbiotic bacteria, free-living alpha, proteobacteria that were incorporated into ancestral eukaryotic cells approximately 1.5 billion years ago. They retain their own circular DNA, a small genome encoding 13 proteins essential for the electron transport chain. They retain their own ribosomes that use bacterial-type protein synthesis, sensitive to bacterial antibiotics, and distinct from the ribosomes the host cell uses. They retain their own double membrane system. The inner membrane, where the electron transport chain runs, is the original bacterial membrane. Every cell in your body contains hundreds to thousands of these ancient bacterial residents. They provide approximately 90% of the ATP the cell uses. Without them, complex life as it exists, animals, plants, fungi, every organism larger than a bacterium, could not function. The energy budget of a eukaryotic cell exceeds what any non-mitochondrial energy system can produce. The symbiosis made complex life possible. When the cell measures mitochondrial membrane potential and flags a failing mitochondrion for recycling, it is managing its relationship with a symbiont older than every animal that has ever existed. Voltage measurement, PINK1 accumulation, Parkin-mediated ubiquitination, autophagosome engulfment. All of this is the eukaryotic cell deciding which of its bacterial guests are still functional and which should be replaced. Mitochondrial inner membrane potential that PINK1 measures is generated by electrochemical physics identical to what drives bacterial membrane potential in free-living bacteria today. The cell is checking whether its ancient residents are still running their original electrochemistry. The ones that are not get recycled. FAST, that triggers enhanced mitophagy, is accelerating a 1.5 billion-year-old quality control relationship between host and symbiont. The person who fasts for 16 hours is not doing something new. They are activating a maintenance program older than multicellular life. The voltage surveillance and the autophagosome recycling are one system. There is another, operating through completely different physics, addressing a completely different problem. Macroautophagy, the pathway described above, the pathway the Nobel Prize recognized, engulfs entire organelles and large protein aggregates in bulk. A second, completely separate system operates continuously, independent of fasting, degrading individual damaged proteins one molecule at a time. This is chaperone-mediated autophagy, CMA. CMA works differently. A chaperone protein called OSHC70 recognizes damaged or misfolded proteins carrying a specific amino acid sequence motif, K-FARCO, and escorts them individually to the lysosomal membrane. There, a receptor protein called LAMP2A unfolds the damaged protein and threads it directly through the lysosomal membrane, one molecule at a time, for degradation inside. No autophagosome. No membrane bending. Individual molecular threading. CMA handles approximately 30% of cytoplasmic protein degradation under normal conditions. The K-FARQ motif it targets is present in roughly 30% of all cytoplasmic proteins. The cell has pre-tagged nearly a third of its own protein inventory for monitoring. The surveillance is built into the molecular structure of the proteins themselves. Well, and this matters specifically for the person over 65. CMA activity declines dramatically with age. LAMP2A levels in the lysosomal membrane fall as cells age, reducing the throughput of this continuous quality control system. The proteins that CMA would have caught and degraded individually begin to accumulate. They aggregate. Those aggregates become targets for agrophagy, the autophagosome-based clearance of protein clumps. But agrophagy is a slower, less efficient system for handling individual misfolded proteins than the molecular threading of CMA. Difference is analogous to the difference between picking up individual pieces of litter as they fall versus waiting until they accumulate into a pile and then sending a truck. CMA is the continuous pickup, efficient, targeted, handling each misfolded protein as it appears. Agrophagy is the truck, effective for bulk clearance, but too slow and too resource-intensive to handle the continuous trickle of individual misfolded proteins that thermal noise produces every second. Accumulation of protein aggregates in aging cells, including the amyloid and tau aggregates implicated in neurodegenerative disease, is partly a consequence of declining CMA throughput. The continuous, one-at-a-time quality control that kept misfolded proteins from accumulating in younger cells is running at reduced capacity in older cells. Fasting upregulates macroautophagy, the bulk system. It does not restore CMA, the threading system. The two pathways address different problems through different physics, decline at different rates, and respond to different rates, and respond to different interventions. Any account of fasting that treats autophagy as a single process is missing this distinction. Think about what this means for the fasted brain. After approximately 18 to 24 hours of fasting, liver glycogen is depleted and the liver begins producing ketone bodies. Primarily, beta-hydroxybutyrate, from fatty acid oxidation. Beta-hydroxybutyrate crosses the blood-brain barrier and fuels neurons directly. That much is widely discussed in fasting content. What is almost never mentioned, beta-hydroxybutyrate is simultaneously a signaling molecule that modifies gene expression through a mechanism completely independent of its role as fuel. It is a histone deacetylase inhibitor. It blocks the enzymes that remove acetyl groups from histone proteins, keeping chromatin in a more open, transcriptionally active state. Genes whose expression increases under beta-hydroxybutyrate-mediated histone modification include BDNF, brain-derived neurotrophic factor, the primary molecular driver of synaptic plasticity and hippocampal neurogenesis, and genes involved in oxidative stress resistance. Molecule that fuels the brain during fasting is simultaneously reprogramming the brain's gene expression toward growth, plasticity, and resilience. It is doing pharmacology and providing calories at the same time, through completely different molecular mechanisms running through one molecule. For the aging brain, where BDNF levels decline naturally, where synaptic plasticity decreases, where the hippocampus is losing volume at a measurable rate per decade, the beta-hydroxybutyrate signal is not merely interesting. It is activating the molecular program that counteracts the specific neural changes of aging. The fuel source the fasted brain runs on is simultaneously the signal that drives the plasticity the aging brain is losing. No pharmaceutical currently available produces this combination of effects: metabolic fuel and transcriptional reprogramming. Through a single endogenous molecule, the body produces on its own when food is withdrawn. Cognitive clarity many people report during extended fasting. The sharpness that arrives after the initial difficulty has a precise molecular basis. Beta-hydroxybutyrate opening chromatin and increasing BDNF expression in neurons that are simultaneously running more efficiently on ketone oxidation than they were on glucose. That dual function, fuel and transcriptional reprogrammer running through one molecule, is the finding that sits at the center of the fasting research for me. Because it means the brain's response to fasting is not deprivation, it is activation. The molecule the body produces when food is withdrawn is simultaneously powering and upgrading the organ that will decide when to eat again. One more thing, what is settled and what is not, because the distinction matters for an audience that will check. What is not settled, the clinical optimization of these mechanisms in humans through specific fasting protocols. Osumi's work was conducted in yeast. The extension to human clinical outcomes, how many hours of fasting produce clinically meaningful autophagy enhancement, whether intermittent fasting prevents specific diseases, whether the benefits of short fasting windows are equivalent to longer ones, requires inference from mechanism, and remains an active area of investigation. Walter Longo's fasting-mimicking diet trials at the University of Southern California produced the strongest human clinical data to date, showing improvements in biomarkers of aging, inflammation, and metabolic health. But on protocols more demanding than standard 16/8 intermittent fasting, the 16-hour window that popular fasting content promotes may or may not be sufficient to produce the level of autophagic induction that the mechanism predicts. Because the threshold varies between individuals, varies with metabolic health, varies with age, and has not been measured with the precision the mechanism deserves in large human trials. Mechanism is real. Optimal human protocol is still being defined. Both are true. Only one is settled. The person who tells you that 16 hours of fasting will definitely clear your damaged mitochondria is extrapolating from yeast and mouse data to a human protocol that has not been validated at that level of specificity. The person who tells you the mechanism is not real is ignoring a Nobel Prize. And then, the second law. Second law of thermodynamics does not negotiate. Every ordered structure in the universe moves toward disorder unless energy is spent to maintain it. Your cells have been spending that energy since the moment they existed, maintaining membrane potentials, refolding proteins, measuring mitochondrial voltage, threading damaged molecules through lysosomal membranes one at a time. bending lipid bilayers into closed spheres around failing organelles. Fast does not introduce a new process. It shifts the balance of an existing one, from building outward to repairing inward, from suppressing the recycling machinery to releasing it, from accumulating damage to clearing it. The mTOR switch flips. The autophagosomes form. The voltage surveillance clears the backlog. The damaged mitochondria are replaced by new ones. The misfolded proteins that escaped CMA are engulfed in bulk. The cell that was running maintenance in the background while prioritizing growth now prioritizes maintenance and suspends growth. The physics reverses direction. Now, consider what modern eating patterns mean for this switch. A person who eats from 7 in the morning until 10 at night, 15 hours of feeding, 9 hours of overnight fast, never crosses the mTOR threshold. 9 hours is insufficient. The switch never flips. The autophagosomes never form at enhanced rates. The flagged mitochondria accumulate another day. The misfolded proteins that CMA missed remain in the cytoplasm. The cell runs another 24 hours in growth mode, accumulating the damage that repair mode would have cleared, because the feeding window never closed long enough for the phase transition to occur. The food is not harmful. The growth signal the food delivers holds the recycling brake in place, and the brake stays on as long as the food keeps arriving. This is not an argument for any specific fasting protocol. It is a description of what the physics requires. The threshold exists. The switch is binary. The cell cannot run enhanced autophagy while mTOR is active, and mTOR remains active as long as amino acids are present above the threshold concentration. The minimum duration required to cross the threshold in humans is approximately 12 to 16 hours. Variable between individuals, variable with metabolic health, variable with age. But the threshold is real, and the physics on either side of it is different. Nobel Prize was not awarded for discovering something exotic. It was awarded for describing the mechanism by which every cell in every body that has ever existed maintains itself against the most fundamental law in physics. Fasting does not damage your cells. It is the condition under which your cells do the work they were built to do. That is the physics.